This is a production of Cornell University.
So I really appreciate the opportunity to talk about the Life Sciences Core Laboratory Center. In terms of my background, I'm a molecular biologist. I came to Cornell about 5 years ago now from the Harvard Medical School, where I helped set up and run the first Genome Center at Harvard. So my expertise is in genomics, part of the Human Genome Project, the Mouse Genome Project, a lot of whole microbial, whole genome assembly type projects, as well as in translational research. So coming to Cornell was really eye-opening to me in terms of all the types of applications that you can do in both agriculture and veterinary sciences. I'm going to be talking about the various technologies in the Life Sciences Core Laboratory Center, as well as some of the ways you might be able to access and know about some of the other shared research resources at Cornell. So I'll start off with an introduction to the Life Sciences Core Laboratory Center, which for short we typically just call the CLC. So it's currently composed of 7 life sciences core laboratories. It's open to all investigators at Cornell University and to outside investigators. It was established a while ago as part of the New Life Sciences Initiative. It's a part of a New York State designated Center for Advanced Technology, and it's administered by the Institute for Biotechnology and Life Sciences Technology. Joss Rose is the director of the what's typically called the Biotech Institute. Now, the mission of the center is to promote research in the life sciences with advanced technologies in a shared resource environment. The center includes fee-for-services research, technology testing and development, and educational components. So these are the 7 core facilities that currently compose the center. So we have a genomics facility. We have there a number of first-generation sequencing instruments, the ABI 3730 capillary electrophoresis instruments. We're doing more than 500,000 samples a year for that. And I should say the mandate of the center is to be of university-wide value. So that means that, as you all know, Cornell president, we've got the Ithaca campus with a big focus in the agricultural and veterinary sciences, the medical school campus, the Weill Cornell Medical College in New York City, and the medical school campus in the Middle East in Doha, Qatar. And our center receives samples from all 3 campuses. In addition, as I had mentioned, we're a New York State, part of a New York State designated Center for Advanced Technologies, which means New York State companies that use us get a discount that's transparent to us. It's subsidized by New York State. And in addition to that, we provide resources and services for about 100 academic and commercial institutions in about 40 states in the United States and about a dozen countries outside the United States. So that's the scope of our operations. So in addition to the first-generation sequencing, we also have a number of second-generation sequencing instruments, and I'll talk a little bit more about that in a minute. But we have the Roche 454 GS Flex and the Illumina GA2X and Illumina HiSeq 2000 instruments. We also have a number of genotyping instruments, the Sequinome Mass Array and the Illumina Bead Express. We have a number of different microarray-based platforms, from Affymatrix, from Illumina, from Nimblegen, and from Agilent. So in addition to the genomics facility, we also have an epigenomics facility. Now, the epigenomics facility is really the first example that I know of a true cross-campus core. A lot of the infrastructure is located on the medical school campus, and we also have a lot of the infrastructure located on the Ithaca campus. So this core provides the IP, the immunoprecipitation for ChIP and ChIP-seq, and also provides a sample preparation for any type of meth-seq type of application. So in addition to the genomics and epigenomics facility, we have a proteomics and mass spec facility. So this facility provides a number of different mass spec instruments. We have a ABI 4700 MALDI-TOF, and we have a number of electron spray instruments, including the ABI 4000Q trap, the water synap, and the latest Orbitrap from Thermo, the Velos. So this core is doing protein identification, post-translational modification, base studies, and small molecule characterization. In addition, we have a very high-end imaging facility. So this facility provides everything from conventional confocal fluorescent microscopy. It provides whole animal fluorescent imaging, high-resolution ultrasound, and both micro and nano Ct. And I'll go into a little bit more detail about that as well, particularly how that can impact some significant projects in plant biology. And I should say we're also working on building up our own multiphoton microscope. So to provide support for all these cores that can give you a real fire hose of data, we have a very high-end bioinformatics facility. We call it the Computational Biology Service Unit, or the CBSU. This core has a lot of infrastructure. We've got a cluster with more than 1,000 CPUs. We have a very close relationship with Microsoft. We're one of only 10 worldwide Microsoft-designated high-performance computing institutes. So the core has leveraged Microsoft funding into building an open-source portal for life sciences application in a high-performance computing environment. And I'll talk a little bit more about that, but basically we have more than 52 life sciences applications in this portal, including next-generation sequencing pipelines that's available to everyone at Cornell. The other area of expertise of this bioinformatics core is building up basically bio-IT LIMS, information management systems that combine all the different types of information you might get from any of these basic resource portals, research type of instrumentation, and combine it with the clinical, with the phenotyping as well. And the bioinformatics facility has also had a big focus now in terms of training and workshops to provide the tools for faculty, students, and staff to utilize the analytical tools. We also have a very high-end bio-IT facility. That's Bioinformation Technology Facility. So this core provides all the laboratory information management systems, the LIMS, for all these cores for tracking of sample submission, process tracking, and data return. And the core also provides fee-for-services for outside academic groups, such as biomedical engineering and other academic departments at Cornell. And finally, we have an advanced technology assessment effort, which is really a way of saying, how do we bring in the new toys to Cornell? I'm director of operations of this whole center, I'm responsible to be sure everything is working smoothly, and I'm also director for advanced technology assessment. So in both my areas of expertise and other areas, and in very close collaboration with the directors of all these cores, we try to determine what are the right new platforms to bring to Cornell. So we basically listen to you. We listen to what faculty say they need to be at the cutting edge of research. We try to identify the appropriate platforms, to bring them in for testing. If they look appropriate for our high-throughput production pipelines, we try to identify sources of funding to acquire these new platforms. And once we have them, we try to further kick the tires to optimize applications on these new platforms, as well as develop new methods and new techniques to more fully utilize and optimally utilize new technologies. So basically, the Core Laboratory Center provides support for everything from genes to proteins to phenotype, all tied in together with the bioinformatics and bio-IT. Now, most of our cores are located in the biotech building. That's the genomics, a part of the epigenomics, the proteomics and mass spec, the bio-IT and the advanced technology assessment are all located in the biotechnology building. Our bioinformatics facility is located in Rhodes Hall. It's hosted by the CAC, the Cornell Advanced Computing Center. And our imaging core is a lot of the technologies in the basement of Weil Hall. It's in a vibration-free section of the basement. And we also have various confocals and other instruments over by the vet school, some of it behind animal barriers. So one of the things we try to do by leveraging having so much multidisciplinary, multi-core expertise all under one virtual roof is really integrated multidisciplinary support for shared research resources. So for example, our next generation sequencing is located, as you might imagine, in our genomics facility, and they're responsible for sample preparation and data generation and first pass data quality analysis. But if we have any types of projects which involve epigenomics or high-end bioinformatics support, or really developing new types of applications, our epigenomics core, our bioinformatics core, our bio-IT, and our advanced technology assessment cores can all get involved in terms of the support and operations of this instrument. And I should say we really sort of have a systems biology approach to the support of research projects. So if there's projects that have data that includes not just genomics, but proteomics and imaging data, at the point of project design, we would bring in all the appropriate core directors to really talk about how to appropriately integrate all these data streams into one type of structure which is appropriate for bioinformatics analysis. And in support of that, as I mentioned, our bioinformatics core has a real expertise in developing integrated databases for SNP genotyping, gene expression, DNA sequencing, proteomics data, imaging data, and it was all integrated with the phenotypic data that you might require. And they developed a lot of this expertise in support of large-scale national plant projects, and have leveraged that all the way into supporting clinical-based, human-based projects just by building HIPAA compliance, where obviously the plants don't care about privacy, but humans do. So I'd like to talk a little bit about each of the cores and start off with the genome. So the types of sequencing technologies that we have in our core include the ABI 3730XL. This is a 96 capillary array electrophoresis instrument. It's the basically the gold standard instrument for first generation sequencing using Sanger chemistry, and was the instrument that actually enabled the Human Genome Project and a lot of other types of whole genome assembly type projects. So the types of parameters that I look at when trying to compare these types of sequencing technologies are read length, number of reads in parallel, the number of bases you get in the full run, the runtime per full run, the raw data storage needs, and the cost per full run. Now there are other parameters as well. How many samples can you run? How much multiplexing can you do? There are a number of other very important parameters. But just in terms of these very basic considerations, the ABI 3730XL produces about 750 bases per sample. You get 96 reads in parallel. It takes about 2 hours, so in about 2 hours you get a little bit less than 100,000 bases. You can run this 24/7. And the amount of raw data storage needs is in the megabyte range. And the cost here, depending upon whether or not you're a small-scale academic core facility or a very large-scale genome center, ranges anywhere from $50 to $500 for a full run. The very first next-generation sequencing technology that we brought into the core was the Roche 454 GS Flex. GS referring to genome sequencing. Currently, well, this is actually outdated, this slide. We currently can get 420 bases, the average read length, but we are upgrading to the latest long read reads chemistry on this instrument And that will enable us to do average read lengths of anywhere from 650 to 750 bases, which really brings it into the read length range of Sanger chemistry. You can get about 1 million reads in parallel. With about 400 base average read length, you get about more than 400 million bases per run. Right now, we're getting about a gigabase per run, a billion bases. It takes about 8 to 12 hours for a single run. The raw data storage needs are in the gigabyte range, and the cost for running a full run, depending upon the application, is in the range of now between $5,000 to $8,000. The Illumina GA2X is an older next generation sequencing technology. It was originally called the Celexa instrument. And we have one of these instruments. The upgrade to that is the Illumina HiSeq 2000, and we have two of these instruments, one which was just installed about 2 weeks ago. So with this instrument, you can get about 150 contiguous bases. You can also do paired end or mate pair type of reads where you're reading from the same fragment from both sides. You get more than 1 billion reads in parallel. Currently we get about half a terabase worth of bases per run. Internally at Illumina, they're getting almost a terabase worth of bases per run. It takes about a week, depending upon the applications and the actual read length that you want to do and such, anywhere from 4 to 10 days to do a full run. And the amount of information that you need to store is actually in the gigabyte range. It actually creates terabytes worth of data, but it doesn't save everything. The data that you really need to save is in the gigabyte range. And the cost for running this is in the range of $5,000 to $10,000 per run. Now, the types of applications you can do on these next-generation sequencing platforms range the gamut of just about everything you can imagine, from whole genome sequencing, both de novo assembly and whole genome resequencing, resequencing of PCR applicons or other targeted regions of the genome, low-frequency mutation detection, genetic variation, including both SNP genotyping or copy number variation, gene expression analysis, including whole genome RNA profiling. And I should say this is particularly useful for looking at degraded RNA from stored samples. And also gene regulation-based studies. Whole genome small RNA discovery and quantitation, genome-wide measurements of protein nucleic acid interactions, which is typically called ChIP-seq, and genome-wide DNA methylation profiling, what's typically called METH-seq-based applications. On the 454, this is a picture of the instrument. This is essentially one of the more expensive type of confocal microscopes you can get, and I'll be happy, I won't get too much into the exact what is the basis of each of the technologies, but I'll be happy to answer questions about that or talk about that offline. This is a lot of fun in terms of how they got to these types of throughput in terms of number of bases. But essentially, here you can see that the mean is about 420 with a spread in terms of the actual read length that you'll achieve. With the newer long read technology, we're expecting the average mean to go about 700 bases. And this type of technology has been extremely important in de novo assembly type of applications with the many non-typical plant and animal model systems that the genomics core at Cornell processes. Just to give you an idea, compared to the traditional type of workflow that you would need with Sanger-based chemistry with the first generation sequencing, on the 454 you can get your data in about 3 days, 2 days worth of wet bench work and 1 day in terms of data generation. And just to give you an idea of this type of impact, about 5 years ago I was involved in a whole microbial genome assembly of a virulent strain of Pseudomonas. a bacterial genome, about 450 million base genome. We dedicated one ABI 3730XL to this project. It took us about 10 weeks to get 10x coverage. And the cost of this project, including library prep and data generation, was about $50,000. Now, we can do this same project for the cost of less than $1,000 in less than a day on our current next generation sequencing platforms. And that's why it's a no-brainer why a lot of different types of projects are being applied to these new technologies. And the technologies are developing very rapidly. We have both the Illumina GA2X and the newest iteration of that, the Illumina HiSeq 2000 in the lab. We have two of these units. The HiSeq just came out as a commercially available project at the beginning of this year. Basically, the difference between these two instruments, which are all related to density of information and speed of scanning and such, allows you to get anywhere from 5 to 10 times the information off the HiSeq than you can get off the GA2X. So you can get about 50 million bases of data off the GA2X. Again, this slide is already outdated. We can get about 500 billion bases off the HiSeq 2000. And there's also a real need for doing an integrated approach with many projects in terms of using multiple types of instruments to support a single project. Just as an example of this, I'd like to just point out a high-density scorable markers for maize trait dissection project, which is I've been involved with, which is the lead PI in this is Ed Buckler with the USDA ARS here at Cornell, and Doreen Ware over at Cold Spring Harbor. So basically, the idea of this project was to try to pull out a couple of million SNPs out of maize. At that time, there wasn't a maize genome available in the public domain. The way we went about this is we did a restriction digest type of approach to reduce complexity. So we got about an 80% reduction of complexity that way. And we use these methyl fractions first on the 454 to build up essentially reference scaffolds that we could then use for the much cheaper, much faster Illumina-based runs. Very much, this was a very successful project and was the basis for the technology that Ed Buckler's group has come out with, which is called genotyping by sequencing. And currently, using this type of approach, we're able to multiplex 384 samples per flow channel on an Illumina HiSeq. There are 16 flow channels on the Illumina HiSeq. So we're doing 16 times 384 samples to do genotyping, which reduces the cost of genotyping to about $8 per sample. This has been such a successful approach that the Institute for Genetic Diversity, which is also under the Biotech Institute, is now creating a service dedicated to this. And we have right now about a dozen plant-based projects and about 8 vertebrate-based projects using this approach, which really brings down the cost of genotyping to enable types of research that just weren't possible before. So in terms of what this type of technology can really do, the Illumina HiSeq gives us a real dramatic increase in the cost, in lowering the cost of per base and in increasing the number of bases per run. So right now, what that means that you can do a human-sized genome on this instrument in about a week at a cost, a consumable-based cost of about $5,000. So this is, basically, this charts the cost of doing a human genome, which you typically do at least a 30x coverage with this type of short read technology, the cost per each year to do this. And you can see that it's been decreasing dramatically. And it's not just doing whole genome types of projects, but you can do, as I mentioned before, targeted genomes, just look at targeted regions of interest. You can multiplex smaller genomes. And you can also do not just genome assembly, but for example, the types of expression studies that you would previously would have really done on microarrays. So you can do with the Illumina HiSeq up to 200 samples for a 2-day run at a consumable cost that's basically equivalent to prior microarray types studies. So the challenges to implementing this type of new sequencing technologies, really, there are many of them, but one of the most obvious is the large and rapidly increasing amounts of data. On the left hand here is the number of samples that have been processed by our sequencing, our genomics core, since it was established in 2002. And this is in a linear scale on the left over here. On the right is the number of bases per year that the genomics core has been putting out. And this is a geometric scale, so a logarithmic scale. So as you can see, we constantly increased the blue here. The light blue is the first generation sequencing. And you can see that the next generation sequencing technologies here in red and green on these bars have enabled orders of magnitude more data. In terms of what the core is putting out. And if you combine the linear scale of the number of samples with the logarithmic scale of the number of bases, it's very easy to deduce that what we're really doing here is getting a lot more information per sample. So next, I'd like to talk a bit about our Proteomics and mass spec facility. So we have a number of different tools for sample preparation and separation, including a number of HPLC units. We have 2D gel systems, and we also have robotics for gel spot and gel elution for the 2D gels. We have a number of tools for data generation. As I mentioned earlier, we have a MALDI TOF, which is an ABI 4700. And we have a number of electron spray instruments, including the 4000 Q-trap, the SYNAP mass spec, and the Orbitrap from the Orbitrap Velos from Thermo. And we provide a number of different bioinformatics tools for analysis, including gel image analysis software, search engines, including Mascot and others, and we have LIMS for data storage and data return. This is our 4000 Q-trap. Here is our MALDI TOF. This is the SYNAP, and this is the instrument that we all really love. We got this in the past year, the Orbitrap, which gives us orders of better resolution and sensitivity than any of our prior instruments. About it in comparing these instruments, here on a very general type of axes is the price is the y-axis, and capability and performance is the x-axis. The MALDI TOF is still used, but it's no longer the the instrument of choice for many types of research projects. The 4000 Q-trap and the SYNAP provide very good data, but in terms of capability and performance, the Orbitrap is really pushing the envelope. And there are a number of different considerations we look at when we compare these types of instruments and determine which one is the appropriate one for any particular research project. And the types of parameters we look at are sensitivity, resolution, accuracy, quantitation, scan speed, mass range, and dynamic range. For any particular project, we'll sit down with you and go over which is the right platform for the goals of your particular research project. So the core provides a number of different types of services. We provide biomolecular separation and sample preparation. both for 2D gel and HPLC. We provide protein ID. We do comparative proteomics, both with 2D gels and iTrack. We do protein characterization, doing post-translational modification-based studies, as well as looking at other types of amino acid modifications. And we do small molecule profiling and quantitation. We provide help with the data interpretation. We also provide training on various types of techniques. The core has been very, very active in implementing new technologies. And this is just a timeline in terms of the new technologies that the core has provided over the past 5 years, going everywhere from 2D gel to iTrack to targeted PTMs, and most recently IMAC and INTEC protein MS analysis. Now, I'd like to also talk a little bit about our imaging facility. So our imaging facility provides a large range of different types of technologies, everything from conventional confocal fluorescent microscopy to whole animal fluorescent imaging to high resolution ultrasound. The thing I'd like to point out is the latest technology that we've implemented, which is both micro and nano CT. So this is the same type of CT when you go to a hospital to get a CT scan, but of much higher resolution. So the types of data sets that we've been generating includes everything from vertebrates to invertebrates, plants, electronics, fossils, anything related to life sciences types of applications, as well as non-life sciences materials sciences type of applications. So our micro CT capabilities provide a lot of different support for a lot of different types of research projects, including paleontology and botany and obviously horticulture. The advantages of this type of technology is it's non-invasive, non-destructive. It provides both 3D and 4D data sets. By 4D, I mean over time. And you can get direct in vivo measurements. So just to give you an example of this, and I hope this works. Okay, let's see if I can get this to work. Sorry. So this is a three-dimensional reconstruction of Snapdragon. So I consider this both some of the most image-intensive, informative, and artistic type of data that we put out in our core center. So, and this is the type of technology that we've been looking at from things you wouldn't imagine, looking at fossils and seeing the internal structure of tribolites. We've been doing it through tomato and not needing to cut into the tomato and just getting a full three-dimensional, very high-resolution structure of the tomato. So these are the types of applications that we can do in our imaging core. So I'd also like to focus a little bit on the types of services and resources that our bioinformatics facility can provide. Again, we call this our Computational Biology Service Unit, and it's typically called just the CBSU. So our project workflow for a lot of our projects and all of our cores is very, very simple. It's investigator initiated. We do start up a project. We put the sample data into our laboratory information management system. We do quality control on the samples that are submitted. In this case, I'm saying sequencing, but for any of these cores, we can do the sample processing and put the data into our data pipeline. And then do various types of data analysis and quality control on a daily, weekly, monthly, and project basis. And that data can then be distributed back to the investigator and also sent to the bioinformatics core for further downstream chewing on the data, and that again can be distributed back to the investigator. So in more complex detail, just in terms of just for sequencing, we really have a number of different ways of integrating various laboratory processes with our laboratory information management system, and the investigator can then access this data at various points. So our LIMS system provides support from everything from data archiving to data analysis, through data tracking, and as well as data generation. And for example, the way our investigators can interact with this, if you provide samples, for example, for a next generation sequencing project, and as I said, it can take a week or more to generate the data off the instrument, and there can be a processing queue for that as well. You really want to know where your samples are in that processing queue. And we got a lot of feedback from faculty about the need for that. And we've implemented various ways now that faculty can go to our website and see just for their own samples where they are in the processing queue and get a very rough estimate in terms of what the data return time and such. Our bioinformatics score provides a lot of different types of bioinformatic pipelines. For sequencing, it includes everything from base calling, production QA/QC, sequence alignment, and data visualization. And all of that goes into various boxes in terms of analytical tools, from genome and targeted region resequencing, RNA-seq, ChIP-seq, or custom types of projects. Now, one of the things that, well, I should say that these bioinformatic pipelines are actually available directly to investigators. And the way that's done is through what we call BioHPC, which is a computational biology application suite for high-performance computing. It's, again, this is already outdated. Here I say we have 48 software applications. We're up to more than 52 software applications that we're currently providing support for, including data mining, population genetics, phylogenetics, protein structure prediction and modeling, and actual next-generation sequencing analysis pipelines. Actually, all the data that's generated by our genomics facility for next-generation sequencing automatically goes to these pipelines, is analyzed and distributed to the investigator using these tools. And we have tools available that the investigators can go back and reanalyze. The types of applications that are on the BioHPC, as I said, include a wide range of applications, and here I just list a small number of them. And we basically put on the applications that our research community says would be useful. So if some of these applications could be homebrew types of software packages that faculty here are developing, and the other of them are commercial or freeware type of applications that the general community uses. Now, another big focus of our bioinformatics facility is in education and training. We have a number of different workshops covering practical aspects of using computational biology applications in a Linux type of environment. We have Linux for biologists, parts 1 and 2. We have one just using the BioHPC software. And we have how to use the interface. So the workshops are focused on practical applications of particular techniques and algorithms in computational biology. Last year we started off with just a general one, for example, for next generation sequencing, and we had about 160 students, staff, and faculty attend. And the feedback we got is that folks wanted more in-depth focus on particular topics. And we've responded with that, for example, by having a RNA-seq workshop recently. And just last week we had a genotyping by sequencing workshop. And we have more coming up. And you'll be able to find out about those by just going to our website. And I'll give you that information later. Training is conducted using the BioHPC Lab. The Hpc Laboratory is actually a collaborative result of the CBSU from the Core Laboratory Center, the 3CPG, the Cornell Center for Comparative and Population Genetics, and the CVG, the Cornell Vertebrate Genomics Group. So, and this has been a very, very useful collaboration. All the workshop materials, including lecture slides, exercise walkthrough, and the data are available online. So if you missed any of these, you can see the materials. And for future ones, you can look at the material before the actual workshops. I'd also like to talk a little bit about our advanced technology. So when we try to think about what new technologies to bring in, from my viewpoint, we really look at it from three different levels. Do you want to be cutting edge? Now, being cutting edge means that you really need to be able to bleed a little bit and not die from it. What we're talking about here is very early stage implementation of emerging technologies. And folks like the Broad or WashU or other large-scale genome centers are really excellent at doing this. The Broad, for example, I believe, has a room where they've chained down a half a dozen engineers from Life Technologies and don't let them go, or from Illumina, I should say, and don't let them go in case any of the instruments break down. Another way to think about this is being an early adopter. And by this I mean early stage development of new technologies where you have an instrument that doesn't break down every 2 days. But when the vendor is releasing it, it really hasn't been optimized in terms of the best way to do the existing applications or really been explored in terms of the full range of new applications you could put on this. And that's actually an ideal sweet spot for our Core Laboratory Center. We do a lot of this, both in collaboration with instrument vendors as well as with faculty on this campus and other campuses of Cornell. And the third level is really the level of a conventional user, where you want an instrument that your most important consideration is that it's giving you good, high-quality data with the instruments never breaking down. And that's the main consideration of a lot of core facilities, typical academic core facilities. You really just want to be able to robustly generate the data. So when we look at advanced technology assessment, one of the big focus areas is on third-generation sequencing technologies. One of them that we've been really focusing on is the PacBio technology from a company called Pacific Biosystems, which is actually a spin-off from Cornell. This technology was developed here. Watt Webb was one of the major developers. Harold Craighead, who is the head of the Nanobiotechnology Center here, was one of the developers. So it was just commercially released recently. You can get a read length on this instrument, on average read length of 1,500 contiguous bases, with the 95th percentile going out to about 3,000 contiguous bases. And it's possible to get even more by pushing the instrument to get as much as 10,000 bases of contiguous read. Again, already this slide is outdated. We now get about 75,000 bases reads per run in parallel. So you're getting about 75 million bases per run. It only takes about an hour or so to do a run with a reagent cost of about $100. So it's very cheap, very fast to run. It's, in comparison to the massively parallel second-generation sequencing technologies that we have here, you can think of this as having some ideal applications that will enable new types of research projects that weren't really possible before. These types of long read lengths, for example, are ideal in terms of looking at SNVs, structural variant components. By that I mean insertions, deletions, duplications, and inversions, which are very difficult to see with small reads, with short base reads. And this type of long read technology is also very good to do in a hybrid sense with the shorter reads to add information to allow assembly. I should say this technology is also, the PacBio technology is advancing rapidly. By the end of the year, the average read lengths are expected to be about 3,000 bases, with the 95th percentile going out to 6,000 to 9,000 bases, and with contiguous reads being able to be pushed out to maybe 15,000 to 20,000 bases. So we currently have a PacBio at Cornell. It was received about 2 months ago on the Weill Cornell Medical School campus in New York City, and it's being set up as its own sequencing core, and we hope that, but it was brought together by a group of investigators that will get primary access, so it won't be a traditional type of core access, but any excess capacity will be available to the community at large. And if folks want to utilize this, you can just get in contact with me, and I'll be happy to put you in touch with the folks down at the medical school that are to help make sure that it will be an effective core. And we have a number of different efforts to try to bring in the PacBio to the Ithaca campus. The types of applications that you might be able to do in plant biology, well, I could go on, but on and on about that, but I think you guys could tell me much more in terms of the types of unique applications. Just very briefly, the way this technology works is that you have a nanofabricated hole on a metal film that's on top of a silica base, and the pore width size is only enough to allow epiluminescence through there. You have a polymerase that's attached to the bottom of this pore, and you're reading the strand of DNA as it goes through. The DNA has, each of the nucleotides has a base at the very end of the, a fluorescent dye marker at the very end of the phosphate chain, and that is cut off after incorporation. So at the end you have a natural strand of DNA which doesn't have any of the types of tertiary structure types of problems that you can get with other types of incorporation of dye terminators, for example, in Sanger-based chemistry. The very nice thing about this technology is that besides putting in that since you're using a polymerase, what they have found is that you can detect modified bases by the amount of time that the nucleotides actually hang at the point of incorporation inside the polymerase. So basically you can use kinetics to detect modified DNA bases, and that's the basis of detection for simultaneous DNA sequencing and methylation-based detection. And you can detect not just methylated bases, but any type of base modification. You could also, instead of putting down a polymerase, put down a reverse transcriptase and do direct RNA sequencing. So instead of as you do with next generation sequencing, where you do you take your RNA, you convert it into the complementary DNA and run that on your sequencer. You can do direct sequencing of that RNA transcript, which means that you have fewer steps where you can introduce artifactual polymerase or reverse transcriptase. You could put down a transcriptome, a spliceosome, or a ribosome. And so you can look at splicing, or you can look at protein sequencing, peptide sequencing. So there are a lot of different types of applications. I should say currently the commercially available application is direct DNA sequencing, but these other applications have already been published and shown in proof of concept, and we'd like to get our hands on this instrument to really push these applications in collaboration with faculty and with the nanofab folks and other folks at Cornell. I should say that since you're doing true single molecule sequencing on this instrument, for standard sequencing where you're just doing one pass sequencing, you're only getting about 85% accuracy. But the sample prep is basically you create a circular structure, and you can do multiple reads of the same circular product, and in that way increase your accuracy to close to levels of other types of sequencing technology platforms. Another very new sequencing technology that's come out is the ion torrent. This is the first true non-optical sequencing technology. Essentially, it's built on a computer chip, which is pictured over here. This computer chip has nanofabricated, massively parallel pH meters on it. At the bottom, you're essentially doing sequencing within each well, and you're detecting the release of the hydrogen ion at the time of base incorporation. The nice thing about this technology, since it's based essentially on CMOS technology, it has very fast cycling times in terms of development. This technology came out less than a year ago with 100 base reads. It's now doing 500 base reads. It's doing about 10,000 reads in parallel. At a cost for a single run, which takes about 2 to 4 hours, reagent-based cost of about $500. And the instrument itself is less than $100,000 as compared to the Hpc, the Illumina Ga, or the PacBio, both of which are about $750,000. So this is a very, very nice new platform that we're hoping to bring in here as well. And we're talking to faculty about who's interested, who might be able to help us acquire this, and we're looking at various grant funding mechanisms as well. I should say, again, down in the medical school, they've just received an ion torrent that's available, again, to anybody at Cornell, so that if you do want to do a first-pass test of this technology, you are able to send samples down to the medical school for that. I won't go into big detail about this, but essentially what we're talking about is in terms of the types of genomic technologies, we have first generation, a number of different second generation technologies, and we're trying to bring in a number of third generation technologies. And the potential projects using these emerging sequencing technologies run the gamut. Everything from application of sequencing by genotyping to breeding, conservation, and global species and population surveys, sequencing of individual human genomes as part of personalized medicine, de novo global gene expression analysis based types of projects, identification of microbial diversity for agricultural, environmental, and therapeutic goals, and things that folks haven't yet done but are thinking about. For example, DNA computing, manipulating DNA libraries for highly parallel computations. So, the second generation sequencing technologies are really looking at doing, the goal which of these technologies was doing a mammalian human-sized genome at the cost of $10,000 to $100,000, which is about 4 orders of magnitude less than the Human Genome Project did for their end goal. Third generation sequencing technologies have the real potential of doing a human mammalian-sized genome at the cost of $1,000 or less, and that price point is being reached very rapidly. There are a number of different approaches for that. I mentioned the PacBio, which that technology is based on what's called the zero-mode waveguides, which are those nanofabricated pores that I had mentioned. But there are other approaches as well. For example, nanopores, pulling DNA through a pore in a silica base and reading it as it goes through. Now, at that level of, that has a lot of challenges. At that level of detection, your detectors can interfere with your detection. And also, the speed in which DNA can translocate through a pore can be very fast and a challenge to single base resolution. But a lot of clever folks are coming up to clever solutions to this. One of the most prominent companies that are working on this is Oxford Nanopore. And eventually, either in the coming year, definitely within the next 2 to 3 years, this will be a very, very viable and very, very exciting technology that will be commercially available. So our problem is, how soon will we have to buy our next new toy? And what new challenges would these new platforms present? Now, finally, I just want to end up talking about finding information about core facilities. In the ARC Core Center, we do a number of different things. We have our website. We have we try to give talks at these types of departmental seminars. We run training workshops. Each of the cores has their own training workshops. A number of the cores have user group meetings. And we have technology-based seminars. For example, we have a Life Sciences Advanced Technology Seminar Series where we try to bring in folks, either vendors or folks using new technologies, to make to bring these new paradigms and new technologies research community. We send out user surveys, and we have a lot of faculty involvement. Each of our core facilities has a faculty advisory board that provides feedback as to the value and the future directions of our cores. And we currently have more than 54 faculty from 20 departments and 6 colleges at Cornell combined on these various faculty advisory boards. We also have a number of educational workshops. I had mentioned about the bioinformatics core workshops, including the next generation sequencing workshop and the bioinformatics core Hpc laboratory. Our Proteomics and Mass Spec core runs a Proteomics workshop and is also has meetings of the Cornell Proteomics program. You know, as I'd mentioned, we have an advanced technology seminar series. We have various user meetings for our genomics core, as well as a list server and Confluence site where users can share, not just as an email you know, discussion, but share software, share publications, and share protocols that they're developing. And I had mentioned the Cornell Proteomics program, which is supported by our Proteomics and Mass Spec core. And our imaging core has the big group, the bioimaging group, and they have a seminar series associated with that. We try to bring in, have presentations by all the core directors. In September, we had an expo where we brought up all the cores from the medical school and had a combined poster expo with all the cores from the Ithaca campus. We're working with Vivo so that, which most of you are probably aware is a search engine for Cornell. So we're trying to make it so that if you put down genotyping, you get not just the faculty that are working with genotyping, but all that have genotyping resources and services and where their locations are and such. So this is still a work in progress, but we do have, Vivo does currently do some of this. And I wanted to also mention all the cores at Cornell, and this is a representative but not exhaustive list of the cores. I've talked a lot about our Life Sciences Core Laboratory Center with our 7 cores, but there are other cores as well that have either departmental focus or a school focus or a single campus-wide focus. And those include the Nanobiotechnology Center, the CNF, the CCMR, as the flow core in the vet school. There's an NMR core, a statistical consulting unit, and a number of other shared resource services and centers located on Ithaca, as well as a number of different core facilities, about 14 down at the Cornell Medical College. The Cuttery campus has been increasing the number of cores until recently. The date palm genome assembly, as well as in medical-based types of projects. They also now have a proteomics and a bioinformatics cores as well. And there are various library resources as well that help you identify these types of core resources. In terms of finding out cores outside of Cornell, I just want to point out that there's the ABRF, the Association of Biomolecular Resource Facilities, has both white pages and yellow pages that identify more than 250 core labs that are members of this International Association of Core Facilities. And there are a number of registries of core facilities, besides the ABRF, the Vermont Genetic Network, and AMDEC, which is a consortium of 26 institutions in New York State, all have their own core facilities databases to help you find out if if Cornell doesn't offer it. If Cornell doesn't offer it and you need it, you should be coming to us as well and letting us know. We have a process whereby faculty can request new cores to become part of our center, either existing cores, departmental cores that might have a value as a university-wide resource, or new cores based on new technologies. And obviously, in the current economic environment, we're limited in terms of the number of cores we can bring under our umbrella. but we do need to hear from faculty as to what would be the best thing for us to do. And finally, I want to say, the whole idea of running cores, offering cores, is to network with faculty, staff, and students. That's why I'm here. And as nature has taught us, networking can provide very, very many productive results. So I'd like to end up with acknowledging the folks at the Core Laboratory Center, all the Core facility directors, including Peter Schweitzer, the director of our Genomics Core, James Van E, the director of our BioIT Core, Shen Zhang, the director of our Mass Spec Core, Yarek Pilar, and Becky Williams, the director of our Imaging Core. We have primary faculty advisors for each of.
This has been a production of Cornell University, on the web at cornell.edu.